COVID-19 Vaccine Recommendations by Julia and Jordan. Currently, many different scientists and companies are working towards a COVID-19 vaccine. The current 44 possible vaccines can be split into 7 different platforms listed on the screen. The manufacturing and other key considerations of each platform were compared to help determine the most successful vaccine.
An inactivated vaccine is typically created by growing and incubating the virus in eggs, then killing it with heat or formaldehyde, and finally purifying the inactivated viral product. This is a long process because it requires waiting for the virus to grow, and the process becomes reliant on egg availability for the incubation steps. This platform calls for multiple doses and an adjuvant addition after purification and a batch reactor to increase the body's immune response. The platform has presented reduced immune response for some vaccines in elderly and obese people, which make up our higher risk group for COVID-19. However, the platform has been around for years and been used for influenza vaccines, among many others. Therefore, it has a fine-tuned process and can produce higher yields. Therefore, it was our initial recommendation, prior to realizing that the manufacturing facilities would need Biosafety Level 3 clearance, because the process requires handling the entire coronavirus. Requiring this clearance would make the timeline longer, especially since these facilities are scarce.
Non-replicating viral vectors use well-established inactivated viral vectors, like the adenovirus, that can't replicate to express virus proteins in the body to receive an immune response. They are produced using vector propagation. This is when packaging cell lines release RNA-containing vectors into a medium, so only the RNA and the elements needed for gene expression will be part of the vaccine. This is typically done on a smaller scale in a lab using roller bottles for the bioreactors. This process can be scaled up, but it lowers the yield of the process overall. Since the vaccine allows for antigen production to occur inside the body, only one dose is required. This allows for more people to get the vaccine. But if a person injected has been previously exposed to the viral vector used, frequently a common cold virus, the body could attack the virus and render it useless.
Replicating viral vectors express a well-established virus to incite the body's immune response. Since the vector hasn't been edited, it acts more consistent with the expected behavior of the virus, giving the body a more complete immune response. The process typically consists of the removal of certain glycoproteins, then vector growth in cells. A measles vaccine was created from incubated, embryonated chicken eggs, or more specifically their fibroblasts, that were grown in roller bottles, infected, incubated, disrupted, and then purified. The production yield is entirely dependent on the harvesting method used for the creation process. This vaccine only requires a single dose due to the antigen production inside the injected organism. However, if a person has previous exposure or pre-existing immune response against the vaccine platform model, the body will attack the vaccine and render it ineffective.
The RNA vaccine platform injects mRNA from the virus into the body where the mRNA is translated into viral proteins that elicit an immune response. The mRNA is produced via in vitro transcription of DNA. This occurs outside an organism and is the same process regardless of what DNA is used, so the process is highly adaptable and doesn't require a live organism. Even though this platform will require multiple doses, each injection can have a lower viral dose because the vaccine will be self-replicating. A lower viral dose vaccine is better for elderly people who have adverse effects to higher viral doses. Since this is a newer vaccine platform, the infrastructure is not pre-existing, but once it is created, it can be used for future diseases due to its ease and adaptability.
In the DNA vaccine platform method, COVID genes are actually amplified, inserted into plasmids, replicated in cells, then harvested and purified. It's a straightforward process that can easily be scaled up. However, it is the only one with an atypical injection. It requires electroporation, or the application of a short, high-voltage pulse that produces aqueous pores in the cell's lipid bilayer to help overcome the barrier of the cell membrane. This could act as a deterrent for people to get the vaccine, and it complicates the vaccine's required infrastructure, lengthening the distribution timeline. The vaccine induces immune responses, but still requires multiple doses. There are no previous DNA vaccines that have been used on humans, though multiple have been used in veterinary medicine. Therefore, there is no pre-existing infrastructure. This method also may not require cold chain distribution.
The protein subunit vaccine platform injects peptides of the virus into the body to build immunity. The peptide and antigens of the virus are grown in yeast cells using fermentation, which is a lengthy process. An adjuvant to boost immune response is frequently needed, but the necessary ingredients are currently scarce. This type of vaccine has in the past had lower effectiveness rates in elderly and obese people, which is a cause for concern since they are in high-risk groups for getting COVID. This platform does have existing infrastructure that is currently being used to produce the seasonal flu vaccine. These facilities could be switched over to make the coronavirus, but this would come at the cost of protecting less people from the flu and could possibly lead to another outbreak.
Virus-like particles self-assemble to mimic the viral structure but use a modified viral genome, making them highly adaptable yet still able to incite the desired immune response. Necessary proteins will express in applicable mammalian systems, specifically the membrane and small envelope structural proteins in COVID-19 vaccines. The method also requires purification in a 6-well-coated plate prior to transfection, then centrifugation, filtration, washing, and lysing steps. Filtration will affect the yield of the process, and additional infrastructure is required to clean and recycle the expensive resin used in the process to make it a more viable option. This platform has presented reduced immune response for some vaccines in elderly and obese people, some of the high-risk groups for COVID. The platform is already in use making human papillomavirus vaccines, so the infrastructure already exists, and no additional outbreak would be caused by halting production, although people do still need this vaccine.
Out of the vaccine platforms, we determined the RNA vaccine to be the most successful. It can be administered at a lower viral dose, which will benefit the elderly. The production doesn't require the live virus, it only requires the DNA. So once the process is in place, it can be used for future diseases and vaccines. Since the infrastructure doesn't currently exist, the vaccine production doesn't pose the risk of diverting manufacturing resources from other vaccines. Out of the 6 possible RNA vaccines, Moderna's was determined to be the most successful since it has the highest cold chain distribution temperature of -4 degrees Celsius, which can be more easily handled by distribution in medical facilities.